It's my pleasure to introduce my friend Adam, who's the head of genome informatics at NHGRI. I had the good fortune that we met up with Adam about in 2012 or so, and they gave us a lot of help in using the long reads that we had started producing off our PacBio instruments, but couldn't really do anything with, and they were developing methods for assembling those into useful things. And at that time, he was at the National Biodefense Analysis and Countermeasure Center, and I proposed doing a long-read goat genome, and somehow Adam convinced NBAC that goats somehow had something to do with biosecurity, so that he was allowed to work on that project.
I didn't really ask.
So he's going to extend that into the future as reads get longer and cheaper and the devices get cheaper.
Great, thank you, Tim. I apologize for showing up last minute today. I'm really not feeling that well, but I'll try and turn on my usual energetic vibe for the next 30 minutes, and if I collapse afterwards, Tim can just shovel me off the stage here. I'm happy to be with you today. My lab is currently located over in Bethesda at NHGRI, and 5 years on from when I first met Tim, we're still doing long-read sequencing and long-read assembly, so that's going to be really the focus of my talk today. When I entered the field of genomics and bioinformatics, it was early 2000s, and the Human Genome Project was officially wrapping up, and this was a cartoon that came out around that time showing the pile of sequencing reads on a table and a bunch of people scattered around trying to figure out how to put the genome back together again. And when we talk about genome assembly, I always like to use this puzzle analogy because it is quite apt. And if you're doing Illumina sequencing, a lot of you are probably familiar with this feeling of being overwhelmed with a pile of very small pieces and trying to figure out how to put them back together again. So computer scientists have been looking at this problem for the last couple of decades, and the way we go about solving this is to build what's called an assembly graph. So I have 2 slides to just give everyone in the room kind of the same basic understanding of what we're talking about when we talk about genome assembly. So if our Illumina reads here are visualized by little strips of paper, it was the best of times. We know what's coming next. Each of those little shreds shares a couple of words with the shred that came before it. So if you have that giant pile of sequencing reads, you look for all pairs of them that share some number of words between them, and you draw an edge to connect those 2 shreds. And if you do that for all pairs of little pieces that were on your table, you end up with a nice looking graph like this, where each of the shreds is one of the reads, and each of these edges means that one shred overlaps another shred by some number of words. And in this Dickens example, you get it was the best of times, it was the worst of times, it was the age of wisdom, it was the age of foolishness. But it's not just one continual string of nodes together. You have these central nodes that have multiple branches coming off of them. And these are caused by repeats in this opening stanza of Tale of Two Cities, where you've got times it was the and was the age appears twice in multiple places in the opening paragraph, and that causes these repeats that cause this branching structure in the graph. So it's not as simple to just take all of the reads, connect them with the ones that they overlap, and get the genome back. You end up with this graph that's not a single walk through, but many possible walks through. And when you extend this to a whole genome, it becomes very complex. Anybody who's done a genome sequencing project knows that you usually end up with these much larger pieces. You don't get the whole genome back, but you get some collection of larger pieces called contigs, and that's anywhere that that graph structure collapses down to a single path without any ambiguity. And so when the assembler builds this graph, it outputs the contigs. These are the pieces that it's confident in its reconstruction because it's not entering into any repetitive sequences. The very easy observation that we made 10 years ago when we started working on long-read assembly was that if you start with bigger pieces in your puzzle, it's a lot easier to put the puzzle back together again. So if we've got the pile of the human genome and instead we order up the 24-piece Dora puzzle, we can all put that together again because now the pieces overlap without any ambiguity, and you don't have 1,000 pieces that all look like the sky. Every piece has a unique characteristic to it, and now you can uniquely reconstruct the genome. This is exactly why you can reconstruct genomes from long reads better than you can from short reads, because there's more context in the pieces. This is a figure from another project that we actually did with Tim and company back around 2015, where we were doing microbial genomes at the time on the PacBio, and we did this as an illustration to show how much better the genome assemblies become when you're using the long PacBio reads. And this is an E. coli genome assembly graph if your read size is 50 base pairs. So if you rewind time maybe 10 years to the early Illumina days when we got 50 base reads, even a simple genome like E. coli that's about 3 megabases long, very low in repeats, you still end up with this tangle of nodes where each of the little circles is a contig, and all of those edges are these ambiguities caused by the repeats. And if you sequence longer and longer reads, if you have 1,000 base reads, say like Sanger length reads, you end up with maybe 10 to 20 contigs, where the only things that are left unassembled in this genome now are the rDNA arrays, because it's a very large repeat that's present in multiple copies in the genome. But our observation at the time, looking through the database of available genomes, is that most microbial genomes, the longest repeat family in any of them, is usually about 7 to 10 kilobase pairs. So if you can get a sequencing platform that generates reads that are 7 kb or longer, you can now reconstruct genomes into a single unbroken piece. So rather than a bag of contigs, you end up with a single chromosome for E. coli in this case. And it just happens by coincidence that 7 kb is also the magic threshold for human genome reconstruction, because it's about the size of the common line elements, which are the most prevalent large repeat family in the human genome. So 7 kb or more average read length off of a platform in genome assembly becomes a much easier problem. And we like easy problems, so we've been focusing on this. The platform that has us really excited over the past couple of years is the Oxford Nanopore platform, because it's capable of generating reads much more than 7 kilobase pairs, but can generate reads 50 to 100 to almost a million base pairs in length. And the instrument is relatively cheap. If you multiplex, I would estimate you could get maybe $100 per bacterial genome at this day and age for a closed genome. And being a computer scientist, I've never owned a piece of lab kit, but this is the first piece of lab kit I've ever bought, because it is relatively simple to run. And one of the reasons it attracted us is because if you put on, in this case, a Bacillus cereus genome, it starts generating sequence data in real time as soon as you put your sample on the instrument. And this is showing on the x-axis here the genome of Bacillus cereus, and on the y-axis this is the coverage piling up, showing the reads being mapped in real time to this genome. And you can see the coverage is now, after maybe 10 minutes of elapsed sequencing, you have probably 5x average coverage of the whole chromosome. And you can make a very strain level specific call onto what microbial genome you're looking at. And really, by kind of a miracle of technology, this is all by threading a long string of DNA through the eye of a needle, essentially. There's a E. coli-based membrane pore inserted into an artificial membrane, threads a single strand of DNA through that, and measures changes in electrical current across that membrane over time that reads off squiggles, essentially, as a current readout. And you can convert that with machine learning back into ACGs and Ts. And so it's, when you stop and think about it, that we can do this is just kind of mind-blowing. And it works. It's kind of fun to think about the dimensions of this, so that the pore they're currently using is this CSGG E. coli membrane protein. And if you scale that up so that that pore is the size of my fist, it's like threading a 3-kilometer rope through my fist and reading that off as a string of DNA in real time, being processed at about 600 bases per second. And that data is coming off onto your laptop and able to be analyzed as soon as it's seen. You hear a lot about deep learning in the news these days for Google image matching and so on. This is actually the technology that's also used to do nanopore basecalling using either recurrent neural nets or hidden Markov models. And as I said, this current comes off, so you've got time on the x-axis and you're reading current on the y-axis, and it gets ratcheted through kind of in discrete intervals, so 1 base at a time, 600 bases a second. You can segment this signal into positions of similar current, run that through some kind of statistical machine learning algorithm, and out the other side you get base calls. And so Jared Simpson at Ontario Cancer Research Institute has a tool called Nanopolish that's able to process this types of data, and Oxford Nanopore themselves have recurrent neural net-based base callers for translating that real-time current data into base calls. So obviously, if you can get 100 kb plus reads, this has huge potential for genome assembly, and that's where my group came in. We were very interested in translating this very unique technology into excellent genome assemblies. But there was a few problems to address, and primarily this was the work of Brian Wallens and Sergey Korin in my lab at NIH. And the first observations are, because you're reading a single molecule in real time and you're using these very elaborate statistical learning methods to call the bases, you end up with actually a very noisy signal. So currently maybe 10% error per base per read, which is much more than we're used to with the Illumina platform, which is well under 1%. You You can also get chimeric reads where one read will go through and another read will follow quickly after it without really leaving enough time to determine that was two separate events. And so you can have chimerisms between your reads, you have a lot of base error, and this needs to be cleaned up. So we spent a lot of time developing computational techniques for removing that base error. Also, you end up with these very long overlaps. If you have a 100 KB read and it's overlapping another 100 KB read, it's a lot of compute to figure out exactly how those reads are related to one another. And so we had to develop fast algorithms for taking that pile of reads on the table and restitching it back together into a genome. And we borrowed techniques that were used in the computer science field, and particularly for searching the World Wide Web over the past couple of years, and adapted those for use in the genome assembly problem. And the results of this first transformation were pretty striking. At the time we started collaborating with Tim, we were still relying on the Celera assembler that was used by Celera Genomics to build the first human genome. And kind of putting duct tape around that to work on the Drosophila melanogaster genome using PacBio sequencing at the time, this took 600,000 CPU hours to assemble that genome. So basically a month of cluster time. With our new assembler called Canoe, with algorithms that are specifically adapted for this type of noisy data, we're able to do the same process in just 500 CPU hours and actually get a better result on the other side. So the data might seem intimidating because it's noisy and because it's new, but with the right algorithms, you can get fantastic outputs. One of those outputs was the kind of first complete assembly of a Drosophila melanogaster genome. So this was using the same inbred line that Celera Genomics sequenced in the early 2000s. We redid that in collaboration with PacBio in 2015, ran it through our assembly process, and for the first time, a genome assembler took a fragmented shotgun genome project and reconstructed entire chromosome arms into single pieces. And so this is showing one of those assembly graphs I was showing earlier, where each of these colored squiggles is a single contig. And so we've got chromosome arm 3R and 3L essentially into single contigs, unbroken from the centromere to the telomere. And that's the first time anybody has done that for a eukaryote, and it was enabled by these long read methods at the time, PacBio. And there was actually new biology to come out of this genome after we made that assembly and released it to the community. The Y chromosome in particular, which was very heterochromatic and had been incalcitrant to study over the years, new genes were discovered and new biology was discovered in that chromosome because of the long read assemblies that we were able to generate. Another interesting application potentially for these long reads is metagenomics. And so PacBio has released a mock HMP, human microbiome sample that's 20 mixed strains. So it's not quite a realistic sample because you've got 20 diverse strains mixed together and sequenced, but without a lot of strain similarity between them. Even still, there's multiple species of strep and staph in here. You take that pile of reads, put it into Canoe, get the assembly out, and each of these little colored circles is an individual genome. So without barcoding, just putting all of those long reads into the assembly process, you get individually resolved genes out the other end. And with the right sample preparation and the right samples, some hope on the horizon that we can take metagenomes and resolve it back into actually assembled strains. So do I think that this long read process can actually solve genome assembly? I do, because we've seen gradual progress over the years. Working with Tim's group and company back in 2012, we first started closing some of the first bacterial genomes where you give it long reads, you get an assembly, and there's no gaps left. You start scaling up to larger genomes. We did the same thing for yeast in about 2014, Drosophila, which I just showed around the same time. And so if you just start extrapolating out, we should have finished human by now, but not quite. But I think hope is on the horizon. And maybe in the next year or two, we'll be able to close all the remaining gaps in the human genome. So to try and achieve that goal, we've been looking at Nanopore in particular. There's two projects I'd like to talk briefly about. One was a collaborative project with Matt Luce, pictured here, actually sequencing on a Nanopore in his hotel room at AGBT a few years ago. And the cool anecdote here is that they have a rapid preparation. That's maybe 5 minutes of library prep time to build one of these libraries for the Nanopore. And it's a transposase-based prep. And so there's a stage where you have to kill the transposase reaction so that it doesn't overly fragment your genome. And they used the hot water bath from their coffee maker to kill the reaction and build this library, and actually got usable reads out the other end. The other project is Clive Brown, who's the Chief Technology Officer of Oxford Nanopore, sequenced himself. And so he claims this is the first time a human being has ever gone from blood extraction, DNA extraction, sequencing all by themselves without any external help. And so he sequenced his own genome to about 60x coverage using these MinION sequencers. As part of our collaboration, we didn't have access to kind of unlimited flow cells like Clive did, so we got up to 35x coverage with the MinION. And you can see here, for Clive's genome in particular, a 19 kb N50 read length. That's about comparable to what we can get on the best PacBio runs these days. And the assemblies are actually better in some cases than what we can get with PacBio. And so I like to show a visual illustration of this, of what the human genome actually looked like in 2001 when it was announced to great fanfare here with Bill Clinton being the MC, as the project is now finished, right? But we all know this wasn't the case. In 2001, the genome actually looked like this, where each of these blocks is a continuous piece of correct sequence, and anywhere the color switches, it's because there was a gap. And so this looks very banded because there's many, many gaps in these genomes. And in fact, it was in many thousands of pieces, and the average Contig size was just a half a megabase pair. If we fast forward to 2017, the advances that we've had over the past 16 years are striking. This is Cliveome, which I just presented, assembled on a MinION. And same illustration, now you've got contigs into single pieces from chromosome centromere to telomere. And the N50 size of this assembly is 30 megabase pairs, as compared to 0.5 megabase pairs for Human Genome 2001. So one person on a pocket-sized instrument sequencing themselves and getting such an improvement is just a fascinating development. But is it any good? The characteristics of this, as I said, are quite noisy, and one of the common mix-ups in nanopore sequencing currently is a purine-pyrimidine mix-up, where it confuses those two, and will often substitute one for another. It also has homopolymer length biases. The best that we can do using machine learning techniques currently is 99.44% identity across the whole genome, which isn't ideal. With PacBio, we can easily get up to 99.99. And so there's still a little bit of work that needs to be done on the machine learning methods to clean up these assemblies and make them very high quality. Here's what the homopolymer calls look like in particular. Here's an IGV plot covering maybe a 15 to 20 base pair poly A region. The original base caller that Nanopore was providing basically gave up after any homopolymer bigger than 5 bases. If it was 10, 15, 20, it didn't care. It would always say it's 5. And so this caused a big error in your process. But they've now improved and released a new base caller called Scrappy or Albacore, which does much better on the homopolymers. And so you're seeing steady progress over the years with this technology. But the most exciting aspect of this is the prospect to get these ultra-long reads, which aren't reads that are 10 kb or 20 kb, but reads that are actually exceeding 100 kilobase pairs. And so Josh Quick, who's in Nick Lohman's group, one of our collaborators on this human nanopore project, developed a protocol where we're able to get an N50 read length of 100 kb. And so people are used to having an N50 contig length of 100 kb being good, but now we can get individual reads 100 kb and longer. And in these cases, half of the data that we sequenced was in reads that was 100 kb or longer. So it's not that we just got a couple of these, it's that half of the data produced by the instrument was in these super long reads. And this enables some really cool analyses. Ada Henri in my group looked at phasing the MHC in the human sample that we were looking at, and we actually got a giant 7 meg contig covering the whole of the MHC locus, both class 1 and class 2. And when you look at that contig and you line the reads up against it, you don't see a whole lot of agreement. Basically, we've made a mosaic of that region because we took both of the haplotypes and collapsed them down into a mosaic representation. And this is common in the assembly process. All assemblers will do this. But these ultra-long reads, because they're so long, it allows you to phase across this whole region using kind of common off-the-shelf tools like What's Hap or HapCut2. And so we applied a phasing process to this 7 megabase contig. And so if I zoom in here now to the very complex class 2 region, which is usually very difficult to assemble in its entirety, we get 2 haplotypes A and B across all of the class 2, including all of the class 2 HLA genes, now essentially phased with reasonably good accuracy across their entire length. And so to do MHC style phasing in the past required trio-based approaches or multiple technologies, but here these ultra-long reads promise to be able to do this de novo from whole genome data, which I find pretty exciting. Another cool application from Karen Miga at UCSC applied to this data was to measure actual telomere length. And so it turns out when you're sequencing a read and it gets into the telomeric sequence, you see the telomeric repeat, but when it gets to these little caps on the end, it exhibits a different signal, so you can actually tell when you reached the cap at the end of the telomere. And so she measured this on all of the reads that started and were confidently anchored in a chromosome end and extended into the telomere. And just from the nanopore reads alone, you can get an accurate measure of telomere length for all of the chromosome arms. And so that's shown here, the average telomere length for this NA12878 sample. And an example here where we have two 200 kb reads that are confidently anchored in the end of chromosome 21 and extend into the telomere and give us a length estimate. So kind of cool. So I do think we're approaching a closed genome. The current human reference, GRCH38, which we've been building for the past 20 plus years, has a 56 megabase pair Contig N50 size. The clivome that I just presented actually has a 30 megabase pair Contig N50 size, so getting close just from Clive's data. And the prediction is if we can collect 30x coverage of these ultra long reads, which is still a little tricky to do from the current protocols, that we would be able to get essentially a reference continuous quality genome straight out of an assembler. So I think what proved true for E. coli will soon prove true for human in the coming years. But the contigs you get out of an assembler don't perfectly equal chromosomes. It's easy for human because we have the answer in advance, so we can just map the chromosomes to the contigs. But if we're doing new genomes, agricultural genomes for instance, we just have a bag of chromosomes, and so what do we do with that? I'm part of this ambitious project called the Vertebrate Genome Project, which is aiming to sequence a reference quality genome for essentially all vertebrate species, but we're starting with the orders to make it a little more tractable. Currently, we're targeting about 250 ordinal groups that we've defined based on a phylogenetic analysis. And the hope is to get contigs that are at least a megabase, scaffolds that are 10 megabases, essentially a 99.99% accuracy, and all of those scaffolds assigned and binned into individual chromosomes. So essentially a reference quality genome for all of the vertebrate orders using the available technologies that I've talked about. One of the first demonstrators for this kind of recipe was Papadum the GOAT, which I heard was talked about this morning. And Brian, who's in the audience, I think it was his daughter that actually painted this portrait of Papadum the GOAT. And in working with Tim and others on this, we applied what I'd like to call the kitchen sink recipe, where we basically threw all of the different data that we could from all of the technologies at this genome, including PacBio, bio-nano-optical maps, high-C, chromatin capture, and Illumina, and we were able to get complete chromosomes out the other end at very high quality and only 680 remaining gaps, which is about equivalent to how many gaps are left in the current human genome. And so really, for the first time, a de novo quality reference genome straight from an assembly process. And this was successful enough that it had people like the VGP think, hey, why don't we do this for everything now that it's possible? And the cost point for building one of these kitchen sink recipes for a 2 to 3 gigabase genome is maybe on the order of 50,000 these days, and dropping very rapidly. The goal is to get these genomes done at $20,000 or $10,000 or less, not counting the informatics. So my summary on assembly is I really do think long reads have solved the haploid assembly problem, and the human genome will have all of the gaps completed in a couple of years. Complete genomes will be the new norm. So if you're tired of dealing with short reads mapped to genomes, being able to read off complete genomes and do the analysis on the actual genome, I think will become the new norm. The challenge are the difficult ones. Diploids, polyploids, metagenomes, and I say populations, in particular the insects. Hard to inbreed, and you can't get enough material generally from one individual. And so you end up sequencing 100 mosquitoes, and then putting back 100 mosquito genomes together is very difficult. So we're still working actively on that, and there's some exciting projects we've started with Tim on methods for being able to confidently assemble outbred genomes from a trio-based approach. And there's a big need for new representations and tool chains for this data type, because as an example, the BAM format that a lot of bioinformaticians are familiar with was broken by these long reads. Essentially, it couldn't handle reads over a certain length, and if you gave it these 100 KB, 500 KB ultra long reads, the format would just crash and it was unusable. And so people are having to go back and retrofit all of these common tools so that they can be useful for this data type. And that's something my group is actively working on. So what's next? Sequencing all the things. So we're an informatics group, and we try to think what's going to be the next need so that we can develop tools and have them ready before we kind of know that we need them. And the two guiding principles that we work with these days is that sequencing is becoming more and more democratized, and sequencing we can basically treat as a free process. We all know the NHGRI cost of sequencing graph. It's just going down and down and down until somebody will pay you to do sequencing, I guess, is the outcome. But when you're thinking about experiments now, it's feasible to say, well, I'm going to do all of the vertebrate genomes to reference quality, and that project really is only maybe $10, $20 million worth of sequencing. And so it's definitely within the realm of the possible now. And so these have some very important outcomes if you take these two conclusions to their eventuality. If really every lab, including computer scientists, can run sequencing now, the era of these giant sequencing centers, I think, is really coming to an end. So, the Broad kind of being the central warehouse for all of the sequencing and data, I don't think is going to happen when every lab has their own sequencer. Why would you want to wait and send it out to be sequenced? And so I think a lot of this now is going to happen in the individual labs, hospitals, and centers. And so there, how do we avoid the data silos where everybody keeps their data and they don't share? And that's something that I'm glad Sean talked a lot about in his talk. And if sequencing is free, it brings up this question that if you had all of the genomes for essentially everything on Earth, what would you do with that you couldn't do before? And how would that enable your research? And if you start thinking in that mindset, you might come up with new approaches to solving problems that we haven't thought of before, because this seemed impossible, but I'm here to say that this is kind of an eventuality that's not too far off. And just to end with this common visualization that I like to show when you're thinking about sequencing as a free process, think about other sensor networks that are free, like this WeatherBug network, where you have cheap instruments distributed around the country that are all sensing in real time particular phenomenon. So here I'm showing a wind map. And so this is all publicly sourced data that's made available by NOAA on their website. You can download that data in real time and build these live views of the current wind patterns in the United States. This only works because all of that data is shared across all of the different cities. And if you had silos of that data and all of the individual labs or centers were only keeping their wind data to themselves, you would miss the bigger picture, that something was brewing inside of those silos and you only see it when you expose the full map and everybody shares their data. And so I made this as just an illustration maybe 7 years ago when we were starting to think about sequencing as a sensor. And then when Nick Lohman's lab took these MinION sequencers to Africa to monitor the Ebola outbreak in real time, they actually saw this. He told me, your wind map made sense to me because we were looking at the Ebola sequences as they were coming in from the field on a country-by-country basis. And as soon as we consolidated that together and aggregated across the countries, now all of a sudden the transmission patterns made sense, because basically, people don't stick to borders. People were crossing the borders and taking different strains of Ebola with them, and that broadened their view of how the outbreak was unfolding. And so by aggregating all of this data, it's really the only way that you can find the patterns, because if everybody's just in their castle looking at their little space of the world, you're not going to be able to see the full picture. And so I really lose a lot of sleep over this, where everybody has a MinION and they're sequencing a lot of data, but they're all just sitting on it. That's a huge potential wasted. And so I'm very interested in how we can figure out solutions to enable sharing of such data. So I'd like to just acknowledge my group and acknowledge our very fruitful collaboration on the Nanopore project and the VGP. In particular, Matt Luce and Nick Lohman have been the guiding leaders of the Nanopore project, and the VGP currently being led by Eric Jarvis in particular, and Richard Durbin and Gene Myers and myself are tasked with trying to assemble the 250 orders if we can get them all done. So thank you very much.
Start with Chris.
Thanks very much. I was wondering how you're planning to annotate the VGP?
So actually Sanger has agreed to do the annotation for us, as well as NCBI. So we're planning to upload each of those 250 genomes when they're kind of validated at a good quality to Ensemble and NCBI, and they've both agreed to run their annotation pipelines on it.
Based on what data?
We're also hoping to collect RNA-seq data for the majority of these that we can, but both NCBI and Ensemble can source related organisms given a certain million years worth of divergence to guide as the basis for that. But for the ones that we can, we'll get RNA-seq data either of the individual that was sequenced or a near neighbor for annotation purposes.
Thank you.
Other questions, comments?
And I'll say, just to follow on to that, NCBI is generally fairly open about running their annotation process if you provide them with RNA-seq and a genome. Through their RefSeq process, they're interested in having reference quality genomes for most species. And so if you have a new good assembly of a species, they'll likely be happy to annotate it. That's what we did for the GOAT genome. Tim.
So you put up my question, like that I had sent to you in advance.
Yep.
But You didn't actually give, I'm interested in, do you have any perspective on, if you had all the sequences in the world, what do you think you could do?
I did kind of cheat out on that. Tim did plant that question. He's trying to manipulate you all. This is generally my answer, because I've been thinking mostly on the microbial side. So if this was in an agricultural setting, I might pitch it in the realm of either pathogens or antibiotic resistance. That if we're sequencing real time all of the herds across the country and measuring levels of antibiotic resistance within the bugs that are just circulating freely in them, then you could visualize patterns of emergence as something starts to come out, see it spread across the different livestock in the US or around the world. In the pathogen sense, when I was at the NBAC, they were very interested in the spread of infectious disease in, say, swine. This virus, started to take out the pig population. Where did it come from? When did it come into the US? How did it come into the US? And they kept trying to analyze this with sequences from like today. And I kept saying, you can't do this retrospectively from sequences you collected today. You needed to have been sequencing 7 years ago when the thing came into the country, so you can figure out where it came in and how it spread. And I think that's something that's really needed, is these sequencing networks, because it builds up. If you're sequencing every day, you build up this timeline of sequence. But if you see something new now, 10 years from now, you can go back in time and say, did I see this anywhere 10 years ago? And where did it come into the country and how did it spread? So that's how I think on the microbial side. On the vertebrate genome side, I don't have as elegant of an answer, but my background is basically in comparative genomics, and so having 250 vertebrate genomes to be completed at high quality, I think would have a lot of power for functional comparative studies, because now you have a lot more power in your conservation analyses. And so I'm interested to dig into that data and see what we can learn. And then as you drill into, say, the ruminants and just do a lot of those, there's different questions you can ask when you go deep inside of a clade versus spread out across the orders that we have. And so there's a lot of little sub-projects popping up around the VGP where they're trying to go deep into one particular order rather than spread across. And I think their comparative genomics will be fun.
I like your in-joke, the minion on the minion.
Yeah.
Because we always call our minion the minion.
Yeah. That was from a metagenomic talk. If we put all of those things into the minion, can we tell them apart?
Anyone else? One of the things that I caught from giving a quick reread of the blueprint here recently is that we were aiming to sequence high priority species, and it wasn't a list of 250, it was a list of, I don't know, maybe 10, and we thought that in the next decade we might get 2x sequence for some of them and 10x for others. So your talk is really a testament to the progress that technology has made over the last 10 years. Thank you. Any other questions or comments for Adam? Okay, thank you very much.
Thank you all.